Hello student, welcome to EPG Pathsala. I am Dr. Ajit Jaswal from Department of Anthropology, Pondicherry University. Today we are going to discuss about the module called as Method of Studying Human Genetics under paper Physical and Biological Anthropology. In this modules we are going to answer some of the important questions like what are the different methods of studying human genetics, why such methods are different from other lower organisms, what is the concept of nature-nurture interaction in human genetics, how can nature-nurture interaction be assist in human genetics, what are the emerging fields in human genetics if there is any, and ultimately what are the scope for undergoing research in human genetics. Let me give you a introductory information for this topic. As we already know that human genetics is mainly concerned the scientific study of transmission or inherited human variation. Generally, genetic information is passed on from parental generation to the younger generation, that is from the parents to the son or to the newer generation or the offspring, and it is expressed in the form of phenotypic expression. Though the genetic is the basic approach to understand the inheritable variation or inheritance variation in all organisms, the method of study the same in human is quite different and unique from those of other organisms. It is due to the fact that human life cycle is inconveniently long and the family size are too small to undergo such studies. Moreover, it is difficult or illegal to procure desired mating as can be done in the case of lower organisms. In this model, some of the important methods for studying human genetics will be discussed. Pedigree analysis. Pedigree analysis is a method to identify the mode of inheritance of any trait based on Mendelian inheritance. It can be defined as the diagrammatical representation showing the inheritance pattern of Mendelian traits. Certain symbols are used in the pedigree studies which are given figure in the next page. This study of the character is started from proband and further traced out those who bear common characters among his or her relative in both preceding and following generation, as many as possible. In the chart, the earliest generation is put at the top, followed by the later generation. Different inheritance patterns such as autosomal and sex-linked, both dominant and recessive inheritance can be determined by analyzing the pedigree. This has been used to understand the inheritance pattern of any genetic disease or traits. The given figure shows symbol used in human pedigree analysis as rectangle which indicate male, oval or circle represent female. The line between oval and rectangle is mating. Then there is a line which reflects the parents and children, the relationship. There is a different line of symbols which are used for dizygomatic, monozygomatic and sex unspecified line. Each and every symbols or diagrams are very specific. There is for number of children of sex indicated, affected individual, heterozygotes for autosomal or recessive, carrier of sex-linked recessive traits, death, abortion or stillbirth, or what is called sex unspecified. Similarly, we can see different kinds of symbol used in human pedigree analysis. The diagram explains a hypothetical pedigree indicating autosomal recessive inheritance. There are certain characters that are unique to each inheritance, and hence it can be used to assess the inheritance. As an example, skip of generation indicate the recessive inheritance, and the female being carrier in X-linked recessive inheritance. Twin studies. Twin studies have been extensively used in human genetics to estimate the heritability of a particular trait, wherein relative contribution of heredity and environment may result in varied phenotypic expression. The method analyzes the genetic contribution of a particular trait as against the environmental factor. Perhaps the first a systematized study on twins was reported in 1924, where genetist Hermann Siemens compared school transcripts of individual versus fraternal twins to understand the genetic influence on trait. Twins can be monozygotic twins or identical twins or dizygotic twins or non-identical twins. Monozygotic twins result from the fusion of one sperm and one egg, resulting into a single zygote which further divide into two at the embryo stage, while dizygotic twins result from two different zygotes, each formed by the fusion of sperm and egg separately. Monozygotic twins are of same sex, while dizygotic twins may be of same sex or different sex. Since monozygotic twins is developed from a single zygote, it is regarded as having common genetic constituent, and dizygotic twins is having a different genetic constituent like that of sibling. Methodology. Methodologically, twin study is a longitudinal study where the researcher compared traits of interest in dizygotic twins regarding together in the same environment or in monozygotic twins rearing apart in different environment or vice versa. It is based on the assumption that the differential traits developed in monozygotic twins reared apart would be affected of the environmental factor. Similarly, the alike traits developed in dizygotic twins reared together would be because of common environmental exposure. One of the important measurements to find out the relative contribution of heredity and the environment for quantitative character is concordance score of a trait. It is defined as the percentage of pair in which both twins express the traits among pair of twins in whom at least one has the trait. Twins sharing a common phenotype are said to be concordant, and as well twins sharing different phenotypes are said discordant. Concordant and discordant we have already discussed. Now let's see. Twin studies have been considered as important method to assess nature, that is genetic, and nurture, that is environment approach. Twins, both monozygotic and digenetic reared apart, shows that the influence of genetic and environmental factor in phenotypic expression. Concordant traits developed in twins reared apart indicate the role of genetic factor. So, heredity versus environment or nature-nurture interaction. Concordance scores percentage of pair in which both twins express the traits among pair of twins in whom at least one has the traits. Twins sharing a common phenotype are called as concordant, whereas twins sharing different phenotype called as discordant. This table shows the concordance value in monozygotic and dizygotic twins. The concordance value of several traits in monozygotic twins are as follows: blood type 100, eye color 99, mental retardation 97, hair color 89, Down syndrome 89, left or right hardiness 79, epilepsy 72, diabetics 65, tuberculosis 56, cleft lip 42. Similarly, we can see the concordance value in the dizygotic twins also. But the concordant value of dizygotic twins were comparatively very less in comparison to monozygotic twins. As we can see in the case of blood type, it was 66, eye color 28, mental retardation very less 37, hair color also very less 22, very less you will found in Down syndrome, that is 7 compared to 89 in monozygotic. Hardiness, that is left or right, is 77, not much difference is there. Epilepsy, high differences we can see that here in dizygotic it is 15, diabetic 18, tuberculosis 22, and cleft lip is also very less in comparison to monozygotic, that is only 5. Karyotype analysis. This karyotype analysis is another important method for studying human genetics. That is, it is also a method to find out the transmission of character from the parent to offspring, or find out the transmission of disease or disorder, or the transmission of several factors from the earlier generation to the next generation. It may have a escaping of generation, may not be. The karyotype is a descriptive chart. The information, the transmission can be explained in the form of a drawing or descriptive chart showing the set of chromosome, including the diploid number of chromosome length and distinguishing morphological feature of some of the chromosome. In this method, sample such as blood, amniotic fluid, then chorionic villi, and any other biological sample containing cells are collected from an individual through different technique. Further, the sample is processed through a number of laboratory procedures such as culturing the cell, that is chorionic villi cell do not need cultured as it contain enough dividing cell for analysis. Cell harvesting, separating and staining the chromosome, scanning microscope slides for observing, etc. Cell images are photographed and manipulated with better resolution through digital computer software. This photographed chromosome image is kept under examination for any sort of chromosomal analysis. The chromosomes are captured while the cell is undergoing cell division, particularly at the metaphase stage where the chromosome become condensed and visible. This figure shows a normal human male karyotype. The first figure shows a metaphase array of chromosomes seen under microscope, whereas the second figure shows the arranged chromosome in a karyotype sheet. Karyotype analysis is another important method for studying human genetics. Karyotype is a descriptive chart showing the set of chromosomes including the diploid number of chromosomes, chromosome length, and distinguishing morphological feature of some of the chromosome. This photographed chromosome image is kept under examination for any sort of chromosomal analysis. This method has been employed to analyze the chromosomal abnormalities, both structural and numerical. And this would be helpful in understanding the prognosis of genetic disorder. For better analysis of the chromosomes are stained with certain dyes that show pattern of light and dark band. Such technique is commonly known as banding technique and G-banding, which is used as a dye color Ginsa reagent being the common one. The method has been employed to analyze the chromosomal abnormalities, both structural and numerical. Numerical abnormality may be trisomy and any other excess or less of diploid chromosome. With structural abnormalities include deletion and insertion of segment, translocation, etc. Such detection of chromosomal abnormalities would be helpful in understanding the prognosis of genetic disorder. Adoption studies. Adoption studies are also other method to investigate the nature-nurture approach. It is well established fact that the adopted child do not share the genetic constituent with the adoptive parent. Comparison of adopted children with biological and non-biological parents can provide an understanding on the heritability of the traits. Concordant character between the adopted children and the non-biological parent would be assessed as the influence of environmental factor. This method has been extensively used in the analyzing the heritability of behavioral traits. Adoption studies have shown that the children born out of schizophrenic parents and adopted by non-schizophrenic parents have higher chance of developing the disorder than the children out of normal biological parents and adopted by the same. This method has some limitation too, as such study require access to good adoption records with stable population over generation. So there are a limited number of situations in which they can be used. Biochemical method. Biochemical genetics is also an important scope for understanding human genetics and its association with certain inherited disorders. Biochemical analysis of protein, enzymes, hormone, etc. are very much important for understanding inter or intra-population variation too. Population affinities and diversity can be estimated from the frequency of the variant of such biochemical marker. Beyond this, biochemical analysis also reveals the fact of inherited genetic disorder. Well-established scientific techniques are very important for the detection of such biomolecules. Quantification of such molecules also provides another facet of biochemical analysis. Data so obtained could be compared with the standard label and excess and short of the same would be considered as threat to inherited health problem, depending on the metabolic or physiological pathway that involve the analyzed biomolecules. Epidemiological studies. Genetic epidemiological studies are again one of the important methods for studying human genetics. As Morton has well defined that genetic epidemiology deals with etiology, distribution and control of disease in group of relatives as well as inherited causes of disease in population. Different study designs have been employed to investigate the genetic epidemiological studies. Screening of risk or mutant alleles that cause the genetic disease is one of the important aspects of epidemiological studies. This can be applied in the detection of hemoglobinopathies or any other genetic disorder and can assess the risk of such disorder in the population as a whole or sex-wise. Certain specific techniques such as electrophoresis are used in the detection of mutation. Sometimes it is referred as descriptive epidemiology in the epidemiological science. It is obvious that collection of biological sample, generally blood sample, is very much needed for any genetic analysis. Association studies are also important facets of human genetic studies. Such studies are carried out to examine the possible association of certain mutation or any other genetic factor in the development of genetic disorder. Generally, in such polygenic inherited traits or disorder, the role of these genetic factors in the disease progression and manifestation may differ from one population to another. Association studies can be family-based or population-based association studies. Case control or case-only studies are some of the models of association studies. In the case control studies, the affected individual or those at risk are considered as case, and those who don't possess the character of being case are included as control. Association of risk, mutant allele or other genetic factor are assessed through statistical approach. In population-based association studies, both case and control are collected from the age-sex matched individual from the same population, as different populations have varied gene pool. But in the case of family-based association studies, control are the family members who are not affected by the traits under consideration. Population genetics study. Population genetics is an important branch of genetics dealing with describing the genetic structure of a Mendelian population. It has been considered as a focal point for evolutionary biology, wherein change in the allele and genotype frequency would determine the evolutionary fate of any population. Moreover, the genetic relationship of inter and intra-population in the form of genetic differentiation and similarity can also be assessed through statistical method. Statistics play a very important role in analyzing and interpreting inter or intra-genetic relationship of the population. Such studies are carried out through certain genetic marker. Genetic marker can be either serological, biochemical, or genomic marker. The allele and genotypic frequencies of these markers are calculated based on the observed frequency of the traits or phenotype, which have been collected from the population through sampling approach. Analysis of genomic marker need sophisticated technique such as DNA extraction, PCR amplification, genotyping, etc. Genotyping is requisite technique even in serological and biochemical markers to identify the variants of the trait. Certain statistical analyses are done from the allele frequency calculated, and the result describe the genetic structure of the population. Secondary information such as population and mythological history, etc. provide an important validation in the finding of the genetic result. Population genetic studies have become a basic substratum for population stratification, which could be used in epidemiological studies. Epigenetic study. Epigenetic analysis has brought a new trend in molecular biological research. Epigenetic deals with inheritable changes in DNA or its associated protein without alteration DNA nucleotide sequence. It has been considered as a dynamic way by which genetic variation interact with environmental exposure across development to produce a phenotype. It is often associated with heritable gene expression. This new approach reassures the so-called concept of nature-nurture interaction. Environment may provide change in the expression of genetic trait, without modification of genetic element of the individual. There are different methods to understand epigenetic analysis. The different methods to understand epigenetic analysis, such as histone modification and DNA methylation. It is well established that abnormal pattern of DNA methylation are associated with various complex disorders. Lower than normal level of methylation are commonly observed in some type of cancer, as removal of methyl group can activate genes involved with cell growth and can increase genomic instability. Depth knowledge in the subject matter would be required to design such studies. It is true that the approach of epigenetic in anthropological investigation is less, but it needs to be given emphasis in the present scenario. In the present day, especially after Human Genome Project, methods for studying human genetics have become wider with the advancement of technology. Earlier, it was more of study of inheritance pattern or heritability. Such an approach has been shifted to technology-based model of study, wherein the geneticists try to analyze the gene and their product. So it can be concluded that with focus with following points like different method, especially in the post-human genome project, inheritance pattern or heritability, gene and their product. Objectives of the study are effect of genetic change in the phenotypic expression, influence of environment on genetic expression, expected to bring a new dimension in the applied aspect. Let's try to sum up the information that we have already discussed in this particular topic in the form of a summary, in the form of a important point, in the form of a recap of information, or we can say that just try to once again recap the information that we have discussed in the whole module. The genetic information is actually is passed from, remember that one, from the one generation to the next generation in the form of phenotypes. The method for studying genetics in humans are quite different in earlier days, but it became more or less same in the genomic era. Human mating cannot be controlled as desired, as we have seen in the other cases. They want to have a good variety, they cross with a good variety. But here, in the case of women, it is not possible because of the number of restriction concern. Pedigree is the important method in which we try to represent these things through a diagrammatic representation, showing the inheritance pattern of Mendelian traits. How the traits or character is transferred from parents or great parents to the next generation or to the further generation. So the transmission of character can be reflected, can be represented with the help of a diagram by having a different, the diagram has a different meaning, different picture, and each picture can be reflected accordingly. It may be relation based or different thing. Different inheritance patterns such as autosomal and sex linked. This autosomal and sex linked, both dominant and recessive inheritance, can be determined through pedigree analysis. The character or the feature, the disease, disorder or the transmission, it may be dominant in nature, it may be recessive in nature. So from these diagrams, we try to analyze the character, because the dominant has a separate characteristic, recessive inheritance has a separate characteristic. We have another method called twin analysis, because there is two type of twin that we have seen, monozygotic twins and dizygotic twins. The twin method analyzes genetic contribution of a particular trait as against the environmental factor. No doubt, environment has some factor, some role into play. That differential trait develop in monozygotic twins reared apart would be effect of the environmental factor because they have a similar genetic material. Similarly, alike traits developed in dizygotic twins reared together would be because of the common environmental exposure. Concordance score can be defined as the percentage of pairs in which both twins express the traits among pairs of twins in whom at least one has the traits. Similarly, the karyotype is another descriptive chart showing the set of chromosomes like 23 set of chromosome, pair of chromosome, including the diploid chromosome length, and the karyotypes is arranged based on the size, length, position of centromere and distinguished morphological feature of some of them, distinguishing morphological feature of some of the chromosome. The karyotyping analysis is used to detect chromosomal abnormalities. The abnormalities may be structurally, maybe because of structural differences or maybe numerical differences. Concordant character between the adopted children and the non-biological parent would be assessed as the inheritance influence of environmental factor. Similarly, the biochemical analysis of protein, enzymes, hormones and so on are very much important for understanding the inter or intra-population variation also. Screening of risk or mutant allele that causes the genetic disease is one of the important aspect of epidemiological studies, where we try to compare between the control and affected with based on certain comparison concern. Association studies are carried out to examine the possible association of certain mutant or other genetic factor in the development of genetic disorder, generally in such polygenic inheritance traits or disorder. Association studies can be family based or population based association studies. The genetic relationship of inter and intra population in the form of genetic differentiation and similarities can be assessed through statistical method in population genetic method. Genetic marker can be either serological in nature, biochemical in nature or genomic marker. Secondarily, information such as population and methodological history, etc., it provide an important validation in the finding of the genetic relationship of interpopulation. Epigenetics, there is another method, another part. It also deals with the inheritable changes in DNA or its associated protein without altering DNA nucleotide sequence. Environment may also provide change in the expression of genetic trait without modification of genetic element of the individual. Thank you.